All right, so just a couple of quick things to talk about before I post the exam 3 tomorrow. Um, I will be posting an extra homework assignment for everybody. Um, this assignment will be worth 5 points, and it's going to be an essay style question like you would see on your exam at the very end. You know, there's 10-point essay questions at the very end, but I'm going to have one on each of the chapters that we've had. So chapters 11 through 15. I'll have one for 11, 12, 13, 14, and 15. So you'll have 5 long answer questions that I want you to answer, and it'll just be an assignment that's due on Canvas. Okay, so I'll send an email out to remind everybody to make sure you get that done, and it'll be due by the date of the exam as well. So I'm going to post that today. And so just make sure you're taking care of the McGraw-Hill lab. But this assignment will be in Canvas. You can access it in Canvas, and you're just going to upload a Word document or fill in that little square for the text box. Okay, so it's not going to be through the lab, McGraw-Hill lab assignments. It's just going to be on Canvas, just like a quiz would be. So you're just going to fill in that text. Okay, and I'm going to give you a heads up. 2 of those questions out of the 5 will be on your exam. I might add more questions actually, just to make it a little more difficult, a little more challenging, so you can have a mix of different questions. But I'm going to pick 2 of whatever the homework assignment is, and put them on your exam. Okay, so I might just leave it as 5. That seems like a lot too. So I'm giving you a big hint. All right, this will be some form of those questions will be on your exam that I'm going to upload on Tuesday. Okay, so the test for Tuesday will be on chapters 11 through 15. I left off the plants, or chapter 16 with the development of plants. I think I'm going to keep them on a separate exam. Okay, because we're going to have animals and plants. Might as well just keep them together. So most of this stuff is going to be DNA technology, DNA, and then evolution. Okay, so we talked about a few different evolutionary processes. So today, I think I'm going to review for the majority, or actually all of the lecture/lab time that we'll have this morning. So you'll have an hour-long recording of review for tomorrow. And then tomorrow during my Tuesday lab lecture time, I'm going to actually lecture on chapter 17 instead. Okay, I didn't want to have 2-hour videos today. I got other classes and things like that. So I didn't want to record for 3 hours just for Bio 20 today. So tomorrow I'll go ahead and lecture on chapter 17, since the majority of you probably don't even care about that chapter yet. It's on the next exam. So I'll be reviewing today from chapters 11 through 15. Okay. So I figured this is a lot of information for this test. So I should cover it as soon as possible before the next exam. That way you guys have a good shot of studying, getting everything down before your exam is due. All right, so that's what it was. You're going to have an assignment uploaded directly to Canvas. Again, just answer those questions to the best of your ability. Answer it like if it was an exam. Open note, I don't care, right? I need you to understand those questions thoroughly. And those are going to be the first couple of questions on your exam that you're going to answer. Okay, so those are your freebies. Okay, so just make sure that you get those done. That way you have 20 points free right away on your exam. Okay, you will have the same style of exam, but again, you're going to be given those questions beforehand, so you should know those quite well. Those are freebies. All right. But all right, so today again we're going to just do a review. I'll be lecturing tomorrow on the Tuesday's lecture time for chapter 17. Okay, so we're changing it up a little bit today. Just going to do the review first. I feel like that would have been easier. All right, so for chapters 11 and 12, so there's a lot of information here, right? We talked about genes, gene regulation, gene expression, and we talked about DNA technology, right? So this is that one PowerPoint slide that had both 11 and 12 on it, right? So you should understand the central dogma, right? DNA to RNA to protein. gene regulation and gene expression, right? So be able to define gene regulation, turning off and on specific genes with a living organism, gene expression, the process whereby genetic information flows from genes to proteins, okay? Um, okay, so patterns of gene expression. Gene regulation in bacteria. So bacteria are a little bit different than eukaryotes, right? So remember, eukaryotes have exons and introns. Remember the RNA that gets, so it goes DNA to RNA. That RNA gets spliced up, right? Gets cut into pieces. The exons get to leave the eukaryotic nucleus and go into a ribosome and then start making protein. Well, bacteria can perform transcription and translation at the same time. So this is something that you do need to understand. This is a different process than what eukaryotes have. So what humans or other mammals or other organisms with nuclei, okay? Bacteria do not have nuclei. They're much more simple. They're much smaller, and they can perform transcription and translation at the same time. So what is that transcription and translation at the same time? So they can actually turn DNA into RNA, and at the same time they're turning DNA into RNA or converting DNA into RNA. A ribosome can plug into that mRNA and start translation. So that would be RNA into protein. Okay, and that occurs at the same time, right? And we talked about an operon. So typically what we hear about in bacteria is a lac operon, or the turning on of a lactose or lactase producing gene, right? So there's a DNA, there's a gene on the DNA or the genome that can produce a protein that breaks down lactose, right? So you've ever heard of individuals that are lactose intolerant, right? We do not produce the gene that would break down lactose efficiently. Okay, bacteria. There's certain types of bacteria that can break down lactose, and the operon, something called LAC-Z gene, the lac operon will be turned on in the presence of lactose. Okay, so here you have an active repressor, right? This, let's just say this is the lac operon, right? The gene that's going to turn on the or this is repressing the gene that produces a protein that breaks down lactose, so it makes lactase, right? This is only going to be unbound from the DNA if lactose is present, right? So RNA polymerase can't come and turn DNA into RNA, right? You need to have some sort of special protein or special activator that can bind to that protein in order to transcribe it, right? So again, lactose is a sugar that's going to bind to that repressor and deactivate that repressor. So you have an inactive repressor. Now RNA polymerase can come, right? And remember, this is going to be very important for bacterial gene regulation because the presence of the sugar will allow for the expression of that gene, right? Because it's a very special sugar. And you only want to produce this protein if the sugar is present, okay? You don't want to be producing lactase or the enzyme that breaks down lactose if lactose is not there, right? And that just makes sense. You're wasting valuable commodities inside the cell. You're wasting amino acids, right? For making these proteins, you're wasting these RNA molecules, right? These RNA nucleotides on mRNA transcription, right, and translation. So it's important that the cell has different regulatory events that occur to prevent the waste of products in the cell, okay? And having these active or these repressors on the genes that can be deactivated with the presence of a specific molecule is important for regulation of bacteria, okay? So here we also have a promoter. It's specific in bacteria. This is going to be a sequence that recruits RNA polymerase, okay? And it just activates, it's going to recruit a very specific protein RNA polymerase to that area, all right? So the operator, this is a sequence of nucleotides near the start of an operon. Repressor, again, we talked about a protein that blocks the transcription of the DNA, okay? So here's that pipeline that we talked about in a eukaryotic cell. So we have chromosome, we have DNA, we have a transcription, right? And then we have introns and exons, all right? And that's going to be very important for eukaryotes, okay? we have multiple layers of gene regulation for eukaryotes. It's very important that us as multicellular organisms have different ways to ensure that we don't form cancers, we don't have misformed byproducts, right? We don't have cells that are wasteful. And we also have differentiation of cells, right? So think about eukaryotes. These are single celled organisms. They can turn on and turn off different genes with the presence or absence of different food molecules or different molecules in general, right? Um, so they can easily change or switch their genes on and off. And they're allowed to do this because again they're single cell organisms and they have little effect on really the livelihood of that cell. Whereas when we're talking about eukaryotic organisms, typically we're referring to larger organisms that the regulation needs to be way more specific and way more honed and way more controlled, right? Because that gives us the ability to form larger animals, larger creatures, larger organisms. Okay, bacteria single cell, they're not going to be as organized, they're not going to have as many genes, they're not going to need to differentiate cells. Okay, whereas in eukaryotes we do, right? Think about it. We have one genome in every single one of your cells in your body, and that includes your liver cells, your epidermal tissue, right? Your eyes, your heart, your lungs. All of those cells contain the same genetic information. Okay, the exact same chromosomes, right? The only difference is in your gametes, right? Your sex cells, and your testes and your ovaries, right? Those cells are going to be different because of meiosis, right? But all the cells that perform mitosis, so all your somatic cells, your cells throughout your body, they have the same exact genetic information. Okay, so you need multiple layers of regulation in order to have differentiation of those cells. That means you need to have cells that turn into your hands, cells that turn into your lungs, cells that turn into your kidneys, right? There are all these different cells. So it's important to have regulation on different levels. Okay, so again, introns, exons, you have packaged chromosomes, you have methylated, unmethylated DNA, which we won't talk about. But there's multiple layers of regulation, and that's what you need to understand essentially. If I give you a question on when comparing eukaryotes and prokaryotes in terms of their DNA expression, okay. Um, so here you have that mRNA again leaving the cell, going into a um, a ribosome. So mRNA into protein, okay? And then you have the activated protein being produced, all right? So X chromosome inactivation. Um, so we do have this calico cat presentation as well. When you have it's something called a genetic mosaic. So what occurs is you do have a bar body forming on your X chromosomes. Okay, so we talked a little bit about genetic inheritance with in Mendelian genetics as well as karyotyping, but um, it, in animals or eukaryotes specifically, we donate one sex chromosome from each parent, right? Well, in the case of females, females actually have two X chromosomes, right? One of those chromosomes needs to be deactivated. So one of those chromosomes is turned into what is called a bar body. Okay, so and it's random, and this is why females typically are not affected by sex-linked diseases like blindness, color blindness in males, right? You typically see males that are more colorblind because they only have one X, whereas females are something called genetic mosaic, which means that they typically will get one bad gene and one good gene, right? And throughout the body, they'll have either the good or bad X chromosome turned off or on. Okay. And in a mosaic, if you don't know what a mosaic painting is or mosaic artwork is, it's little pieces of different painted either porcelain or glass that's put into or makes up a larger image, right? And this is essentially what this is referring to. Either one of these is going to be um, turned off or on. And in this case of the cat, you have either the black or this yellow or orange fur that is going to be the gene's going to be housed on the X chromosome. So either one of these is going to be turned off or on. You can see this cat this calico cat is a genetic mosaic, right? You have black areas on the fur and some areas of orange. So again, you have a mix of which chromosomes to turn off and on, right? And females are like this, okay? And again, this is just showing the regulation of genetic information in eukaryotes. It's much more complex than bacteria, right? Again, you have a large organism. You need to turn off and on certain chromosomes or certain genes in order to have differentiation or in order to have that specific trait shown by those organisms. Okay. All right, the initiation of transcription. So we have a transcription factor, and eukaryotic cell, a protein that functions in initiating or regulating transcription. And transcription factors bind to DNA or to other proteins that bind DNA. Enhancer, again, it's the same thing. It's going to be stimulating that gene for transcription. Silencer in a eukaryotic, so it's a DNA sequence that inhibits the start of a gene in transcription, right? An activator, a protein that switches a gene or a group of genes by binding to DNA, okay? All right, and we also have alternate splicing. So we have introns and exons, right? So introns are going to be clipped out and left inside of that nucleus, whereas exons are going to be spliced together, right? These 1, 2, 3, 4, 5 segments of the RNA are going to be spliced together, okay, or they can be spliced together. And what this means is alternate RNA splicing is you can have multiple RNAs produced from one DNA segment, one DNA gene, right? That's going to be found. And this allows for variation. This allows for differentiation in proteins, right? Multiple outcomes can be produced. And we see this with a few different organisms, but we do see this as well in humans with a few different genes. There's probably genes we haven't discovered yet that also have this characteristic, right? And I talked about antibodies being one of them. We have variations in antibodies where a few variations of a specific gene can be changed and mixed together in order to have genetic variation to bind to different types of pathogens, right? But you can also have variation in proteins this way as well. So different types of proteins can be produced in one gene, right? And different variations of that protein can be produced as well. Okay, so very, very different outcomes can be produced from the same gene. All right, and exons allow for that. Exon splicing or alternate splicing. Okay, it's important you understand that. All right, so signal transduction pathway. So there's a series of molecular changes that converts a signal received on a target cell surface to a specific response, okay, inside the cell. So again, this is going to be that touching or the signaling, and then you have a cascade event. So you have relay of proteins, transcription factor activated. That transcription factor can come inside of that nucleus, bind to the DNA, and then you have the activation of that gene, transcription, and then translation. So a new protein that can react to the stimulus can be produced, okay? And that's generally what you need to understand about signal transduction, right? So you have a receptor starts a cascade event. So you have relay proteins, activation of a transcription factor. That transcription factor can come into the nucleus and start the transcription translation, right, events. And then that new protein will be in response to the molecule that's present here. Okay, so homeotic genes. So a master control gene that determines the identity of a body structure of a developing organism. So in plants, they're called identity genes. Again, these homeotic genes are going to be the genes that are going to be controlling what the cells turn into throughout the body. Okay, right? So effects of homeotic genes. Here we have a fruit fly, and then this fruit fly has an extra pair of legs, right? So this can be a mutation in one of those homeotic genes. Again, very, very important, right? You can have mutations, and sadly, you can have these changes shown dramatically in young, right? You can see children born with an extra limb or extra appendage deformities, right? And this can be seen throughout the animal kingdom, throughout we've seen in bugs or different types of insects, and then it can be seen with mammals and various other organisms as well. But again, any multicellular organism has these homeotic genes that will differentiate, right? You have these eye genes, you have these antenna, you have the thorax, the leg, different types of leg genes, right? If you disrupt these genes, you can have catastrophic events occur in the young. Okay. So we also talked about the basis of cancer. So you have an oncogene, a cancer-causing gene, usually contributes to malignancy by abnormally enhancing the amount of activity of a growth factor made by the cell. So a growth factor, a protein secreted by certain body cells that stimulates other cells to divide. A tumor suppressor gene, a gene whose product inhibits cell division, thereby preventing uncontrolled cell growth. So we talked about tumor suppression before in elephants, right? Elephants do have much more cells or much more tissue than humans do, right? So they're much more likely, in statistically speaking, they're much more likely to have cancer, right? Just because there's so much more cells, there's so much more tissue. But in order to combat the formation of cancer, they have multiple copies of these tumor suppressor genes within their genome. Okay, so it can overcompensate for having so much tissue, right? And again, oncogenes, the controlling or regulation of cell division. That's essentially what cancer is, right? It's uncontrolled cell growth in tissues forming the tumor, and then eventually you have the metastasized cancer cells that are moving throughout the body and causing issues. Again, uncontrolled cell growth takes away from the body, takes away from your nutrient, therefore causing issues in that individual. Okay, so understand that uncontrolled cell growth, and we talked about growth factor and oncogenes, which are going to be very important for the tumor formation, right? So you have a proteoncogene, a protein from that stimuli, stimulates cell division, right? So we can have a mutation within the gene. We have the oncogene, hyperactive growth stimulating protein. Again, you're going to have increased cell growth this way. So multiple copies of this gene. You have normal growth stimulating protein in excess, right? You have mutation within a control region of the DNA, mutated promoter, normal growth stimulated protein in excess. Okay, so again, these are the different ways that a proteoncogene can turn into an oncogene, or a gene that can stimulate extra cell growth. Okay, and again, there's also multiple different factors that can contribute to whether or not a cell can become cancerous, right? We talked about the cell cycle, right? Mitosis, G1 and G2 phases. Those phases can be disrupted as well. So if you have an event that occurs in the DNA where it changes or disrupts that, you can develop cancer as well, not just through these proteoncogenes, right? So a tumor suppressor gene, a normal growth inhibiting protein is produced, right? So this is controlling the cell cycle. Cell division is under control. You have normal cell growth, right? A tumor suppressor gene normally codes for a protein that inhibits cell growth and division. So such genes prevent cancer formation, right? You have a defective non-functioning protein. You have uncontrolled cell division. The cells will not check themselves, right, through interphase, prophase, anaphase, telophase, and then cytokinesis. They can be continually dividing unchecked, given this protein's being produced either not sufficiently or there's a defective protein, right? And then you have cell growth and malignancy shown here. Okay, so DNA technology, a couple of things here. So we have biotechnology, the manipulation of living organisms to perform useful tasks. I believe this is hint, hint, this is something that you have on your quiz that's due. So we have a genetically modified organism, or a GMO, right? An organism that has acquired one or more genes by artificial means. If the gene is from another organism, typically of another species, the recombinant organism is also known as transgenic organism, okay? An organism that contains genes from another organism, typically of another species, right? And this is where we can have issues here. So we talked about having different plants, maybe with a peanut protein, and then giving that plant to the general population. If that peanut protein can trigger someone's allergies, we might have issues with that genetically modified organism, right? We also have genetic engineering, or the direct manipulation of genes for practical purposes. And we do see this quite often now. I performed some genetic engineering for my graduate thesis project. And again, can be splicing or changing certain genes within a cell, okay? And I personally worked with bacteria, so it was much easier to perform than if I was working with a larger organism or an organism or eukaryotic organism for that matter. Again, multiple layers of gene regulation. And here you do have a transgenic um, fish here. So you have fish that are have DNA from a jellyfish, right? And this you show this fluorescent display in this organism now. All right, so recombinant DNA techniques. We talked about plasmids and DNA cloning. Plasmids, remember, they're small ring of self-replicating DNA. Okay, think of this as almost like a computer virus in bacteria. Um, this DNA is separate from um, the genome. Okay, just think of it as another separate, like nucleus almost, or another separate genome they have. It's like a baby genome inside of that cell. I know we talked about mitochondria before having their own DNA, and this is almost similar to that, right? Our mitochondria were remnants of old bacteria that were able to come inside of the eukaryotes, and then we enslaved those mitochondria, right? That were once bacteria. Plasmids essentially do have their same they have a separate set of DNA. They produce a separate set of proteins, and many times we can use these plasmids in recombinant DNA techniques for bacteria. Okay, so DNA cloning, the production of many identical copies of a specific segment of DNA. Okay, and we can also use plasmids in conjunction to perform DNA cloning events. Right. So what you do is you cut the plasmid, and then you insert the genome or different parts of the genome. So here's a plasmid again. You see the bacterial genome here, the bacterial chromosome. It's large, right? You have a lot of genetic information. And then here you have the baby plasmid. Okay, this is just a little bit of genomic information in comparison to the full genome of that organism. But again, this plasmid is going to be important because it may or may not provide it a genetic advantage. Okay, a lot of times bacteria house antibiotic resistance genes on these plasmids. What that means is that they can readily accept this plasmid because it's going to provide them with an extra advantage. Okay, so there's certain species of bacteria that will transfer this to other bacteria. Okay, such as Pseudomonas or Acinetobacter baumannii. And what that means is that this can be given to other bacteria, and they'll share resistance genes, which means they're going to share the ability to be resistant to antibiotics. So think of MRSA, or what we typically hear it called as MRSA, right? Methicillin-resistant Staphylococcus aureus. And those organisms can share those genes and allow for the survival under exposure to antibiotics, right? That's an example. MRSA is just an example, but there's other organisms that do share plasmids. MRSA typically has the MEK-A gene on the genome and will be resistant that way. But again, plasmids are important for bacteria. Okay, so using recombinant DNA technology, we have multiple uses, right? There's insulin, there's different proteins that can help prevent blood clots and heart attack. We have genes that can break down biochemical wastes or biochemical hazards, right? We have genes that are used to alter our crops, right? We can add different traits from other plants onto desired plants that we want to grow and survive. Okay, one of the biggest or the most well-known cases of genetic engineering, one of the first uses of genetic engineering is production of insulin for diabetics, and they produce insulin in E. coli. Okay, so again, E. coli can have this inserted insulin gene in a plasmid. This plasmid can be expressed in through activation of either with a protein or some sort of activation molecule, and then you have the production of this insulin, right? And it's cheap, it's quick to harvest, and you can make a lot of it very quickly. Okay, and again, it saved a lot of people's lives, but again, it also provided a lot of money for big pharmaceutical companies too. Okay, so I think insulin is quite expensive for how much it takes to make it. I think it's um, I don't know, there's issues there. But again, it's very, very cheap to grow E. coli. It's very, very cheap to produce this insulin now. Um, purification, other things that there might be a little more difficulties there, but they've got it down to a science. This is old. This is an old case study now, right? This is very this isn't new, right? They've been making human insulin for a very long time in E. coli. Okay. All right. So gene editing and CRISPR-Cas9. We talked about this as a protective measure in bacteria. I think it was specifically streptococcus species. But what happens is bacteria have this specific Cas9 gene that's being produced, and it can edit out viral genomic information. And what you see now for genetic engineering and genetic manipulation of microorganisms or organisms in general, it's typically exploiting something that that cell has produced naturally in its own environment. So we talked about restriction enzymes. Again, that is produced by bacteria in order to combat viral infections, right? So here you have the Cas9. Again, it's utilizing bacteria to clip out or edit out unwanted genetic information, right, in bacteria. But here we can utilize it in eukaryotes to either edit out or add a specific gene, right? And you have the Cas9 protein here, an RNA guide. Cas9 guide RNA complex binds to the target DNA sequence, so it's a very specific sequence. You can edit this Cas9 protein to have an RNA sequence that binds to your gene of interest, right? So part of the target gene here, Cas9 protein cuts the DNA. Normal copy of gene serves as a template for repair enzymes, okay? And then you have the ligation, and then the normal copy of the DNA can be then inserted or clipped together, okay? But you can either remove or add DNA, and that's essentially what is occurring here. You can edit DNA within a live organism, and that's very difficult to do, okay? It's before Cas9 you were unable to really do this unless you were performing this in bacteria, because then you could have what is referred to as a knockout cassette, or you're going to have other types of viral genomic information or genomic proteins that can be used to clip and cut the DNA in live organisms. But other than that, it's incredibly difficult to edit a genomic information once an organism is alive. Okay. It's much easier to produce a new clone or an offspring, right? But if someone has a genetic disorder, let's say I have a genetic disorder that I want changed, it's very difficult to change all the cells in my body or those target cells in order to amend or fix that genetic abnormality that's causing that disease. Okay, and I think we talked a little bit about this at the end of the chapter with Jesse Ginslinger and his case with viral editing treatments. Okay, and we'll talk about that later. But remember, Cas9 is going to be useful. It's going to be useful protein for editing genetic information in a live organism. Okay, so we talked about in the human insulin being produced in E. coli. Okay, remember this is going to be a plasmid-mediated production of insulin, right? So we have a plasmid that has the information encoded for this insulin protein, and then we shove it in a bacterial cell, and that bacterial cell can then be induced or coerced into producing this insulin, right? Because bacteria don't need insulin. They don't have pancreas. They don't have to control blood sugar levels like we do, right? E. coli, they're single cell. They're not going to have issues with diabetes, whereas humans do. Okay, we have genetically modified goats. What was it that they did? Oh, they did spider DNA within goats. So they were producing spider milk and spider protein within the goat milk. Okay, which is very cool because spider web is, or spider silk, I should say, is very expensive, but it's very strong. Okay, and so it's useful for making various different fabrics. I think it'd even be used for bulletproof vests and things like that. But again, they were able to harvest more of those spider proteins by genetically modifying goats with spider DNA. So you have a spider goat, right? Genetically modified potatoes could survive salty or differential environments from cholera. So cholera is Vibrio cholerae is a type of gram-negative bacteria that causes cholera, right? So the very bad bouts of diarrhea, but they were able to harvest target very specific genes that can help potatoes survive better in different conditions. Okay, genetically modified corn, we can add specific genes that'll protect them from different pathogens, right? So corn borer insect here shown here, we are able to add very specific genes that can protect them from these pathogens. Okay, well, we can have transgenic, uh, just more vegetables and things like that. So human gene therapy is a big one, right? So we talked about a recombinant DNA procedure intended to treat disease by altering an afflicted person's genetics. Okay, so I think we talked about the Ginslinger. Let me see. Oh, we'll talk about that later. But essentially, what happens is we are editing genomic information within a human. Okay, and when you edit genetic information within a human, it becomes very difficult because there's multiple cells that are afflicted with this inappropriate gene, right? So many times you need some sort of vector to induce the genetic change from occurring. You need some sort of delivery device to those cells or that tissue in order to have the genetic change occur. Okay, we'll talk about that at the end. I think the Ginslinger case is probably the best time to talk about that. All right, so PCR, polymerase chain reaction. Remember, it's going to be multiple copies of initial gene segment in a machine or in a very specific format, right? So what you need for a PCR is you need the nucleotides, you need DNA polymerase, and you need your primers, okay? Remember PCR and DNA replication is the same thing, right? You have something called semi-conserved replication. So one of these halves of the DNA is going to serve as a template, okay? So you have the conservation of one half of the DNA. Again, they'll be conserved in the next round of transcript or of the PCR cycling, okay? So what you have here is a thermocycler. You have all of the genetic components housed here. You have your initial DNA segment, and then you have the primers that are going to bind. Okay, so remember we talked about short tandem repeats, identifying the father or identifying the crime scene or suspect to see if that individual was there at a crime scene. So collecting DNA, you have these tandem repeats, which are unique to individuals. Then you can identify who was there specifically based on the genetic information, right? So what you have is these tandem repeats. You have the primers or that genetic information that can be amplified in that tube. You have this gel electrophoresis. So you guys did perform a lab on this, right? Where you can load the dye with the impregnated ethidium bromide gel, right? Remember that DNA fluoresces when exposed to UV light. You have the fragments that are treated as well with restriction enzymes, so they chop up the DNA in very specific patterns, and then you have these fragments that are then produced, right? This fragmentation of these copied DNA fragments, okay? Through this fragmentation process, you can identify similarities, right? You can look for which DNA fragments or which DNA samples are going to be more unique, and then you can tell or determine whether or not the criminal was there. Okay, you can compare the crime scene DNA with the person of interest DNA. Okay, and you can look for it. All right. So viruses and other non-cellular infectious agents. So we're going back to chapter 10. Viruses are incredibly important for viruses, and the discoveries that have been made with viruses are incredibly important with genetic engineering now. Viruses just do a lot of very cool things that we did not know could even be possible, right? We talked about the central dogma from DNA to RNA to protein, right? Well, viruses, we've discovered, have the ability to go from RNA back into DNA, okay? So we do have RNA-based viruses, right? The coronavirus is actually an RNA virus, so their genome, the coronavirus actually has an RNA genome rather than a DNA genome, right? Which which is very unique. It's very different. You don't see animals or plants or bacteria or any other microorganisms doing this, right? It's very unique. Um, HIV is the same way, and we had a lot of new discoveries made with HIV because again, HIV was a big problem. We needed to solve this problem. We also discovered a lot of very interesting things in cellular biology from that virus. One of the things is reverse transcriptase, right? It's a very special enzyme that can convert RNA back into DNA. Okay, and we use this now for different analytic instruments, right? Especially when looking for those special RNA viruses, like um, the coronavirus or like influenza. We have the ability to test for that now because of HIV. Okay, um, so yeah, viruses very important. Okay, so we have adenovirus. So we have a protein coat. We have DNA. We have these spike proteins. Sometimes we do have envelope viruses, which will have an envelope on the outside. HIV does have an envelope. Coronavirus has an envelope, and so does influenza. They all have envelopes. Okay, and those envelopes are going to be the things that bind to the cell. Here's an adenovirus. Adenoviruses will bind through these spike proteins. They can induce the endocytosis this way. Okay. Um, Here we have that envelope. Should I talk about this? No, don't worry about this. I'm not gonna. Yeah, don't worry about it. I'll talk about that later. Okay, bioinformatics. So study. Don't worry about this either. Just know the definitions of this. Okay, DNA sequencing. Again, it's the new type of understanding of genomic information. This is bioinformatics and genomics. Think of 23andMe. Know that our sequencing technology is becoming way better. We're able to look at multiple different genomes or multiple different genes. There is some issues here with ethics, right? We talked about how individuals having control of your or information in general. They can tell whether or not you have certain diseases or you're prone to certain cancers. So it can be easier or for insurance companies to decide whether or not to insure you or whether or not to increase your insurance premiums because they know that you're going to you're probably going to develop colon cancer in the next 20 years, right? Because of your information, okay? Because of your genetics, okay? So yeah, bioinformatics again, uh, that is important. So we have again DNA sequencers. This was not possible 30 years ago. There's no way. The amount of technology now that they have and the new and how quickly they're able to sequence a full genome now is insane. Okay, it took so many years for scientists to discover or map out the full human genome. Now we can map genomes out in a day. Okay, depending on how good your computer is. Okay, um, so 500 million bases in a single 10-hour run, right? So these are high throughput DNA sequencing machines. Very important. So we can compare whole genomes now. We can have this data crunched with supercomputers, right? There's a lot of different things that we can do now. Yeah, so important. So we talked about ethical issues. Um, yeah, we can have transgenic uh, um, pathogens that can be very bad, right? I think I talked about my project where we made we essentially made a super bug that was resistant to every antibiotic that you can think of, pretty much. Okay, so that is a problem, right? We're changing the genetic information of a specific organism. We're enhancing its effects. We're giving it resistance genes. Um, that can be bad if it gets out, right? Especially if it finds a susceptible host. We can have issues. Okay, someone could die. But even more scary is the understanding or the study of these very pathogenic viruses, right? That have a high mortality morbidity rate. Okay, And that can be quite scary if that gets out. All right. So yeah, those laboratories need to be monitored. So GMOs, there's a lot of hate or a lot of misinformation and a lot of I guess prejudice or opinions against GMOs. Most of the canola oil that we have, I think all of the canola oil that we have produced in the United States is genetically modified. A lot of the corn, a lot of the different produce, a lot of different produce items are going to be genetically modified now. Uh, as bad as that is, but it actually helps us maintain a stable food source. There are issues with genetically modified organisms, right? We talked about labeling, or we have, um, oops, I think someone's not muted. There we go. Okay. So we can also have the mislabeling of that food source, right? So let's say we have a genetic or a gene from a peanut plant being expressed in a corn or corn cells, right? You can have issues with allergic reactions that way too. There's also very special chemicals that we use to treat these organisms in order to have the mutations or the changing of the DNA. So it's important to have, I think colchicine is one of them that they use to have polyploidy in plants. So it's important to make sure that you don't give those that fruit out or that produce out to individuals after a treatment. Okay, it needs to be the young, or it needs to be the progeny of that plant that has been treated. Right, you don't want to have these chemicals that are out and being fed to individuals. But again, they've got a lot of this down to a science. For the most part, it's safe. You're eating protein in different types of plant protein, different types of sugars. It's essentially the same. Okay. So genetic testing, we talked about insurance companies getting a hold of your information. And yeah, and that's it for that one. Okay. Let's see, 12, 13. All right, so evolution. Let me see, we'll go through this quickly. So two-part naming. Okay, so be able to define natural selection, a process in which individuals with certain inherited traits are more likely to survive and reproduce than other individuals of that species, right? So natural selection essentially is just referring to the random chance that an individual organism is more likely to survive and reproduce. If you are more fit, if you are better able to survive in that environment, you will survive and produce young. If you're unfit, your genetic information may or may not be passed down to the next generation. Okay, so and essentially that is going to cause those minor changes over time and eventually lead to evolution or different traits in that species. Okay, so evolutionary adaptation, inherited characteristic that enhances an organism's ability to survive and reproduce in a particular environment. So evolution refers to descent with modification, an idea that living species are descendants of ancestral species, right? That were different from present day ones. All right, so we have the proof in the fossil record, right? So we have various organisms that were once there that are not there no more. And we also have organisms that we can compare to these reptiles or these mammals, these ancient organisms, and we are now seeing the descendants of those organisms. Again, looking now and looking at the fossil record, and so on. Okay, so the comparison can be made. Okay, so we have insects embedded. This is just different fossil homologous structures. Okay, anatomical signs of descent with modification. So homologous structures, we're looking at mammals or organisms within that same kind of group or phyla that have similar bone structures or similar structures, but they have different uses of those structures, right? So a whale, very different than a human or a cat, and it looks more like a fish. But we do have the same bones. Therefore, we point to a common lineage. Okay, so we point to a common ancestor. Bat, same thing, right? We have a common mammalian ancestor, and you can see this in the homologous structures between these three organisms, right? We do have a humerus or a radial bone, right, that we can then compare. Um, all right. So fossil record, homology, right? Similarity of characteristics from shared ancestry, so the bone structure. Molecular biology, so we can see very specific gene structures, very specific proteins produced in a very specific subset of those organisms. We also have vestigial structures. So these are going to be useless or unimportant structures found in that organism, and they're going to be remnants of the ancestors, right? So like whale bones will have, or whale organisms, or whales, sorry, I can't talk right now. Whales will have a specific bone in their abdomen that is remnants of a leg, or remnants of a hind leg. Okay. And so now that bone is there, and it's eventually going to be absent as the generations continue. Okay. Um, you also have homologies and embryologies, or homology within embryology. So similar adaptation, similar growth in the embryonic stages that we see. So human versus chicken, right? Very similar. We do have the pharyngeal pouches, postanal tail, right? You have different developmental features that you can see or observe. That's going to indicate a common lineage there. Okay. Embryology is a little bit old, right? We've are a little bit of a older piece of evidence that we really don't use anymore, because there could be multiple reasons why embryos develop similarly. They don't really point to common lineages per se, because a lot of the embryos can look very similar, but they can be very, very different in origin. But again, this is kind of an old school method of comparing and identifying common ancestors, right? Um, Now we use genetics. Now we use DNA. Okay. All right, we talked about a phylogenetic tree. More similar related organisms are going to be closer together on the phylogenetic tree. So natural selection and artificial selection. So the process whereby organisms better adapted to the environment survive and pass on their genetic information, right? That's natural selection. We talked about artificial selection, right? The selective breeding of a domestic plants and animals to promote the occurrence of desirable traits in the offspring. Right. And we as humans are not the best at selecting for organisms. We have different dog breeds. We have different, uh, um, you know, livestock breeds, different types of plants, different types of agricultural products that are being produced now because of or artificial selection. But we still have these issues that can come up, right? Especially we see this with dog breeds, right? Dog breeds have very specific traits, right, that we like, such as a pug or a bulldog, or even like Dalmatian or Jack Russells, right? So you have all these different types of dogs, but they also come with their very specific problems as well, right? Short-nosed dogs or snub-nosed dogs do have breathing problems. Certain dogs are more prone to blindness, right? Labs have bad hips. So those are those artificial selection outcomes that we don't really predict for, right? Nature is the best, typically would, I mean, yeah, it is the best at allowing for the organisms that are more fit to survive and reproduce, right? The more favorable traits will be retained in that population. Whereas when we perform artificial selection, we want those desired traits, right? The snub nose, the short stature, various other traits. But we also select for uh, very, we can select for problematic traits also that we don't see, right? Such as blindness or bad hips, things like that. And then it talks about artificial selection. We have selected for larger fruit, larger produce, right? And if not, the tomatoes would be tiny. Don't worry about that. Overproduction. So this is a good case study, right? Evolution of pesticide resistance in insect populations. So we have, by random chance, these specific insects that are resistant to the pesticide, either through a random mutation or a random genetic mix-up. Okay, all of these organisms get wiped out. The green organisms get wiped out by the pesticide. Only the organisms with that specific trait survive. They will then survive to pass on their genetic trait. And we talked about bacteria in this regard, right? Bacteria have a very fast generation time. That means they're able to reproduce very, very quickly. They can expand their numbers very, very quickly in comparison to humans. So we can see evolution real time with bacteria. And we've seen this um, readily since the 1940s, or the advent of antibiotics, right? So the mass production of penicillin, nothing works or no organism can be treated with penicillin anymore except for syphilis. Okay, so and that's a big indicator of evolution, right? We had to develop new and modern antibiotics because the bacterial organisms that we've been treating have been evolving resistance to all the antibiotics that we've been using. Again, um, bacteria are evolving much faster than we're discovering antibiotics. Insects are evolving to take or to withstand these pesticides, right? You have again these new survivors that are going to reproduce and have new generations that are going to be resistant, right? um, Lice, bed bugs, mosquitoes, again, they're all becoming very, very difficult to kill. They are evolving to deal with the new types of treatments or the new types of chemicals that we're using to combat them. Okay, so artificial selection, natural selection. So we talked about it a little bit, right? We choose here for artificial selection. Natural selection, nature chooses. Okay, genetic variation, gene pool. Mm, we talked about the relative. We've done this in the lab, so you don't worry about it. I won't ask you. Bottleneck effect, genetic drift resulting from drastic reduction in population size. I think I talked about. We talked about cheetahs, and then I think we talked about elephant seals, right? So at one point there was like 10 elephant seals left in the wild, right? Due to overhunting. Okay, they used it for blubber. So during the industrial revolution, the production or the discovery of oil and the use of oil, they stopped hunting these elephant seals, right? But the bottleneck effect essentially refers to the decrease in the population size at such a dramatic level, or such a drastic level, that you have a very small gene pool now. Okay, and this illustration is very good at showing that, right? You only have a few of those organisms surviving, and you lost all of that genetic variation that occurred over time in that population. So you can have very specific traits that are kept in that population that might not have been there previously, right? You might have bred those out after successive generations, right? But you see this as well with the Amish in certain types of genetic disorders, right? If there's such a small group or small population, you're going to have conserved genetic abnormalities within this small population, right? Same thing with cheetahs. So cheetahs have a very low number of organisms in the wild, and there's low genetic variability, so you're more likely to have an extinction event. The elephant seals have recovered nicely. Cheetahs are still suffering quite a bit. Okay. Founder effect. So genetic drift resulting from establishment of a small new population whose gene pool represents only a sample of the genetic variation present in the original population. Right. We talked about squirrels and going on the other side of the Grand Canyon. Right. We also have small isolated populations such as the Amish. Right. And the issue here is if you end up in an island or you end up in a very small conserved area, like the Amish will only have families with other Amish people, other Amish individuals. Um, you see this with certain Jewish populations as well. They're conserving their they only marry within a tight knit community. Okay, so the founder effect essentially is referring to that isolated, controlled amount of genetics being passed around only in that community. So sometimes you'll marry second cousins, sometimes you'll marry or they'll have kids with individuals that are very closely related to themselves, which can be a problem, right? You're going to have those incidences where you do have the homozygous recessive trait being expressed. Okay, you're more likely to have issues. Okay, gene flow, the gain or loss of alleles from a population by the movement of individuals or gametes. So let's say gene flow can be referring to, we can use the squirrel population as an example, right? You have the squirrels that are separated, they're starting a new population. But if a new squirrel gets launched across the Grand Canyon, and then can breed with that population, that he or she, that squirrel, can then have a new mix up, a new set of genetic information from the old population, right? We talked about this with lakes too in Alaska and the northern countries or the northern states, right? They have these large pools of water, but they're genetically isolated. But you're going to have the crossover. Let's say if there was a trench dug between the lakes, or you have a pipe that was inserted that allows for fish or or organisms to go in between those lakes, right? That's gene flow. Okay. Gene flow in pollen. So again, pollen or conifers produce large amounts of pollen into the air. They release that into the air, and you can have gene flow from one very specific population and going to another mountain range if the winds are right. Okay. So you have gene flow there. All right. Let me see. So 13, 14. All right. So origin of species, we talked about natural selection. Um, yeah, biological. I know these definitions. We talked about finches and the diversity among species. So you have different variation within species that allows for traits to be selected for, or desired traits to be selected for. Um, You have reproductive barriers, factor that prevents individuals from closely related species from interbreeding. Many times these reproductive barriers can be broken down in prezootic and postzygotic. So prezootic, again, before the egg gets fertilized, and postzygotic is going to be after the egg is fertilized. So prezygotic, so temporal isolation, mating or fertilization occurs at different seasons or times. So they just don't breed, right? Maybe a wolf, a certain type of wolf will breed in during the winter, whereas the other wolf or the other dog species breeds during the spring. Okay, to have a summer children, right? There's a lot of there can be variation there. Habitat isolation. So again, differences in where the organism lives. Behavioral isolation, right? We have courting rituals. So there's prezygotic rituals associated with birds, right? If one bird species produces or does a different dance than the other, they might not be attracted to each other. Okay. So mating attempt, mechanical isolation. So structural differences prevent fertilization. So maybe their genitalia does not fit right together. Okay. Gametic isolation. Female and male gametes fail to unite in fertilization. So it can maybe the ability for that sperm to survive in the female reproductive tract. It could be the inability for mechanical inability, so inappropriate swimming, inappropriate fusion, right? There could be a lot of different things that can occur at that mating attempt. So post-zygotic barriers. So this is going to be the donkey and the horse making infertile young, right? So that's after the egg is fertilized. You may have non-viable young. Okay, I think a horse and a zebra, I think they produce a sterile young as well. I think a lion and a tiger, the liger being produced, also hybrid, but produces a sterile young as well. The liger is like huge, but um, does not it does not have the ability to breed with other organisms. Okay, so reduced hybrid viability. Hybrid zygotes fail to develop or fail to reach sexual maturity. Reduced hybrid fertility. Hybrid fail to produce functional gametes. Hybrid breakdown. Hybrids are feeble or sterile, right? So you could have heavy mutations or heavy issues with the genetic recombination. Okay, remember the zygote is fused between a sperm and an egg cell, right? And that fusion means that these haploid cells, right? These are half as much genetic information, right? You get half information, half from your mom and dad, right? If you have the fusion of the egg and the sperm, and there's not equal amounts of chromosomes, you're gonna have the reduced viability, and then you also have the infertility. Okay, so one organism has 24 chromosomes, us, right? We have 23. We fuse those chromosomes together, they will not fuse properly, right? You have an odd number of chromosomes and an even number of chromosomes. So therefore you will have mixed matched and inappropriate fusion. Okay. These are some pre-zygotic barriers, post-zygotic barriers. Mechanism of speciation. We have allopathic speciation, the formation of these species and populations that are geographically isolated. Think of squirrel and Grand Canyon. Sympatric speciation, the formation of new species and populations that live in the same geographic area. So now we have niche exploitation. So this, think of the Darwin and the finches. They lived on the same island, but certain finches ate bugs, certain finches ate fruit, certain finches ate seeds and nuts. Okay, so now you have differentiation within that that species, but at the same level. Whereas allopathic speciation is going to be barriers. Okay, polyploidy. So this is a random event that occurs with, especially in plants, but an organism has more than one set of chromosomes, more than two sets of chromosomes rather. Okay, so multiple chromosomes that might prevent them from breeding. Okay, allopathic speciation, right? Squirrels. So you can have geographic barriers, but you can have the reintroduction. This is just showing that. Oops, that's annoying. Sorry, that looked ugly. That was bothering me. So great tree frog, again, polyploidy. This is multiple copies of the chromosomes, more than two copies. Okay, yeah, let's not worry about polyploidy too much. All right, we talked about finches, microevolution. Okay, so macroevolution, evolutionary change above the species level. Examples of macroevolution include the origin of a new group of organisms through a series of speciation events, the impact of mass extinction on diversity of life, and the origin of key adaptations. Okay, so think large evolutionary events. Okay, so mass extinction is one of them. We have bottlenecks. We have drastic weather changes as well. There's a few other things there. But we can see this evidence in the fossil record. That's essentially what you need to know. Think of I can't think of asteroid hitting the Earth, boom, destroying everything. Or a large volcano, okay? And you can see this in the fossil record, right? You can see the ash. You can see the large amount of organism found in that rock. And then you can see the absence of organisms for years or decades after that event, okay? And that can show that can display those evolutionary events, right? So we have different geological time scales, okay? Right, we can see this in the sedimentary rock too, right? The examples. We have radiocarbon dating, right? So this is important, you should know this. A method for determining the ages of fossils, rocks, ratio of radioactive isotope and non-radioactive isotope. Remember, carbon-14 has a half-life of 5,730 years. 1% of all carbon is C14. This is atmospheric carbon. So you get the C14 from the atmosphere. If you're dead, if you're in the ground, you start losing the percentage of C14, and we can measure that by the isotope, right? The radioactivity of that isotope. Okay, we have very special technology, very special machines that can detect C14, and we can measure the relative abundance. Anything less than 1%, we know is going to be quite old. Okay, so older than 5,730 years, right? Talked about plate tectonics, the moving of the plate, Pangea, mass extinction, right? Macroevolution here. Phylogeny, so the evolution of history species. So again, we can see that the skunk and this otter are going to be more closely related. The wolf and the coyote are going to be more related. The outgroup is going to be this panther. Okay, so we talked about outgroups, we talked about closely related species. I will put a question on the exam like this. I'm going to ask you which one is more related to the panther or which one's more related to the skunk, vice versa. Okay, so you should be able to determine that, right? Clades are going to be that specific group. So we talked about the in group and the out group, right? And you should be able to determine this. I'll ask you which one's the in group or the out group. You can tell me this group is the in group here, and the iguana is the out group, not closely related, right? And vice versa. So go over these slides. It's gonna be a simple question. So just be able to identify which organisms are more closely related, right? 3-domain system. We talked about the domains: archaea, bacteria, and eukarya. Okay. All right, that's it for there. Let me see. One last slide. Evolution. Origin of life. So we talked about the chemicals that were in early Earth, joining of macromolecules together, the synthesis. We talked about this very specific experiment by Harold Urey. Again, he had a bunch of different inorganic chemicals that will eventually turned into organic chemicals through this heating process, right? He had electricity, he had water vapor, he had methane, nitrates, hydrogen gas. Then he had shocking events again, and then he had a condenser, and he collected these chemical samples. And what he found, he was able to create all these different amino acids, DNA, sugars, lipids. Well, he was able to create amino acids rather. So he was able to do or create organic compounds from inorganic compounds, and that was a very, very important study that was performed, right? We also talked about meteorites that fell to Earth in 4.5 billion years ago. Okay, and it had 80 types of amino acids, 80 types of different amino acids, right? And that can be an indicator that some of these organic molecules came from space rather than being produced on Earth. There's a lot of different theories that are there. The heavily supported theory is this, though, that primordial Earth had all the components necessary to make all the ingredients to life, and provided those ingredients that were able to be then utilized to make life or create life. So abiotic synthesis of polymers. So again, we have new experiments that are linking polymers or large organic molecules to environmental conditions of the ancient Earth as well. So we have self-replicating molecules. They believe that RNA was most likely the first genetic component that was produced, right? RNA, and then protein, and then eventually DNA was utilized for genetic storage. Okay. Oh, excuse me. So we talk about prokaryotes and eukaryotes, early life, deep ocean vents. Archaea live in extreme environments. Bacteria are everywhere. We talked about different shapes, biofilms, endospores. Go through this. Be able to identify the shapes. Cyanobacteria, again, it's photosynthetic. These are heterocysts. These are nitrogen fixing. Yeah, so go through these. This is just, I think most of it's memorization. Yeah, I'm running out of time. So that'll be the end of the review here. But yeah, pathogen, exotoxin, endotoxin. Just be able to define these terms. I'm not going to ask you too many deep questions, but I'll give you that. I'll ask you a question. That 10-point question will probably be from either 15 or 14. So yeah, I like bacteria, so I'll probably give you a couple questions from here. But again, just be able to define, identify the different types, right? You have green algae, chlorophyta, rhodophyta or red algae, and then bryophyta, which is brown algae. But you should be able to easily identify that. It's not too difficult to tell. Okay. Any questions? Any questions? So again, you're gonna have the at least five free response questions that I'm going to provide you for homework that you need to do within the next few days. And then I will upload the exam tomorrow. Again, you'll have 2 days to complete that. So Thursday, you'll have till Thursday night at 11:59 p.m. Okay, but please do those questions. They're on Canvas. I'll post them on Canvas, and you just need to go in and fill in your answers. 5 points, and one of the or two of those questions are going to be on the exam. Okay, I'm giving you 20 points on the exam, freebies, right? Those are the 10-point questions at the end. Okay, so those will be the freebies. You'll get 20 points of freebie points right off the bat. So please do those. Please study those. That is what I will use on the exam. Okay. So any questions with that? Nothing. And make sure that you're also completing the lab assignments on McGraw-Hill. Remember, this assignment with the free response questions, the exam questions that I'm going to be giving you ahead of time, that is going to be just on Canvas. You enter that just on Canvas. It's not going to take you to the McGraw-Hill site. Okay. So it's going to be like a quiz. You just do it. You just do everything on Canvas. All right. I have no questions. That's it for today. The recording's a little long, so it might take me a little while to upload, but it'll be done by the end of the day. Okay. If not, send me an email. I know some people send me emails about it, so please do if it's not uploaded on time, because sometimes I forget or I think I uploaded it today. Okay. Any questions? Nope. Okay. Well, I will see everyone tomorrow if you decide to show up for chapter 17 lecture. Okay. Again, chapter 17 is not on this exam, so it's just 11 through 15. Okay.